Okay, so this video is going to just go through this slide and the next one. This one is comparing replication and transcription because there's a lot of similarities between the two. So let's go through the stack of cards on the right and see which one it belongs to. So only happens prior to cell division. Well, the purpose of replication is to make a copy of the DNA, and the purpose of transcription is to make a temporary copy of messenger RNA. Replication copies the entire DNA strand, but transcription is only copying a portion, just a gene. So that first one is going to go into replication because it happens before cell division. Uses DNA polymerase. That's going to be replication because it's making more DNA. It needs to make another DNA polymer. Produces a double-stranded molecule. Well, because replication is making DNA, transcription is making RNA, we know the difference between them. DNA is double-stranded and RNA is single-stranded, so that's also belonging in replication. It reads only one strand of DNA. Well, in order for replication to happen and get two identical copies, we need to read both strands. So this one goes in transcription. Transcription is only going to read one side of the DNA. Uses RNA polymerase. This is transcription as well because we're making more polymer or more RNA which is a polymer. Okay, and then occurs throughout the life of the cell. That's going to be transcription. Replication doesn't necessarily happen all the time throughout the whole life of the cell, but transcription does. It has to read the genes and make proteins all the time. Uses both strands as a DNA template. Again, that goes with replication because we're using both sides. We have to make the identical copy. And then produces a single-stranded molecule that belongs in transcription. And like I said, we're going to continue on the next slide as well with the fill in the blank. So for genes to become proteins, DNA must first be transcribed. And then during this process, the enzyme RNA polymerase binds to a section of DNA and assembles the nucleotides guanine, cytosine, adenine, and uracil into a single-stranded nucleic acid.